Hi, my name is Andrew Ta, and I'm a junior at South San Francisco High School, and this is the effect of salt concentration on the germination and root development of radish seed. Whenever I go to the supermarket, I'm in awe of the stands of produce that line the walls. This food doesn't come from nothing, though. We rely on farmers all over the world to grow these fruits and vegetables that feed us. Yet many environments restrict the selection of crops that can be grown, and a large portion of cultivated and irrigated land is experiencing high amounts of salinity, which is bound to increase. We've learned that soil and water salinity is measured in decisiemens per meter, a unit of electrical conductivity, and that soil salinity may impede plant growth. So, we set out to answer this question. How does watering radish seeds with a 7 decisiemen per meter saline solution compared to a 0 decisiemen per meter saline solution promote the growth of the root length over a period of 7 days? We chose 7 decisiemens per meter because it was a testing point where we knew that the salinity wasn't high enough to kill the plants, but it also wasn't low enough to the point where it had no effect on the plant. In our method to answering this question, we placed radish seeds in petri dishes that were watered using either a saline solution or a non-saline solution, and for 1 week, we measured the length of the developing roots in 12-hour intervals. Here's what we found. As seen in Figure 1, over the span of 7 days, our non-saline control group exhibited higher rates of root development than our saline experimental group. We calculated that the saline seeds had a 25% decrease in root length compared to the non-saline seeds. And Figure 2 highlights the noticeable differences between the root length of the two groups. Strangely enough, even though the differences between the two groups are apparent, we didn't discover a significant difference until we compiled the data of all 4 trials. And Figure 3 compares the averages of the root length and standard deviation of the two groups 7 days after being watered. Thus, we rejected the null hypothesis and found a significant difference between the two control and experimental groups. Our findings were similar to that of LF Marcellus, which explored the effects of salinity on seed germination and water and nutrient uptake. However, it's important to acknowledge that while making our saline solutions, our calculations were incorrect. Our solution concentration wasn't correct, but when our results were compared to the article by Marcellus and the research of our classmates, we can conclude that salt stress, specifically at 7 decisiemens per meter, negatively affects the germination and root development of radish seeds. And further research gave insight into how salinity specifically affects plant growth. For one, radish reactive oxygen species, or ROS, overproduce under salt stress. And promoting antioxidant metabolism is one mechanism that plants use to detoxify ROS and tolerate salt stress. Ion imbalances are another factor of high salinity stress in plants. And looking at our proposed salt tolerance mechanism, we can see that these proteins control the flow of orange sodium and yellow potassium ions through the cell membrane. These cation transporters are an essential method of alleviating salt stress by ultimately maintaining a low concentration of sodium within the cell and a high concentration of sodium outside of the cell. By understanding this method of salt tolerance better, we're able to analyze specific genetic differences between salt tolerant and non-salt tolerant plants to promote higher ion pump activity. Through biotechnology, these isolated genes could be inserted into plants, like our radishes, through the transgenic approach in order to create more salt tolerant organisms that would aid agriculture in high salinity environments. Salt stress is a major global issue, and understanding to what extent it harms agriculture, and specifically how it does so, allows us to use biotechnology to increase the viability of crops and potentially other organisms for our benefit. And I want to thank Mr. Ng and the Genentech Future Lab grant for helping conduct this experiment and for funding this study. And I want to thank the audience for sharing your time with me and listening to my presentation. Thank you.